Good morning, everyone. So my name is David, and today I'm really glad to be showing you our database for viruses at the JGI. So today I would like to talk about our user website for the viral database, img-vr. So then we will talk a little bit about the different contents that you can find there, different types of data, different operations that you can perform, which ones are the source of the data, how we collect this data from our metagenomic database, which one are the next steps that we are envisioned just to maintain and to grow the database. Finally, how these resources how this effort that started a year and a half ago has been increased over time. So this is the website of img-vr. So you can find under this URL. So and also you have all the information in a publication recently last year. So basically, we have like a very friendly user interface in which you can find different information about all the viral context that you can find in the database, from isolates to metagenomic viral context, and then a lot of metadata and a lot of information related to the grouping of these viruses. I mean, the different clusters that you can find, habitat information. Based on geographic location, different habitat type information. Also, you can find prediction of the host, and also you will have some resources that will talk about how to download some of the information that we have. So the different data that we have in the database is, so we pull out all the reference isolates from NCBI. We also have a collection of metagenomic viral contexts from metagenomes. We also have a huge collection of spacers coming from all microbial isolates and also from different microbiomes. So we are using this information for host prediction and also for habitat prediction and habitat information. We classified our viral context into groups. So you will see this viral OTU happening here and there. So this is our idea of a viral species, viral genus, a viral population genome, let's say. Then we also have the information of all the different models that we use in order to predict our viral context, all these viral protein families, and also, as I mentioned, some metadata associated with host taxonomy in some cases when we could apply one of these hallmark genes for some specific groups of viruses, and then habitat information and geographic location. So those are the total numbers that we have currently. So I'll talk a little bit more about that at the very end. But basically, you see that since last year, so we increased the database dramatically up to a number that we have now, around 700, more than 700,000 metagenomic viral contexts. And also we have these large numbers for spacers and all the information here. So basically, for example, we have around 5% of all the contexts that we have a host predicted for them. So some of the operations that you can do in the website are these kind of query approaches based on BLAST. So basically, you can blast your sequences, your predicted viral sequences, against any of the data types that we have, isolates or metagenomic viral contexts. And also, you can use the different spacers that we have, storage as a database, to query your predicted context. And finally, this is a new addition that is, so we are really happy that everybody in the community now, they can download easily the data. So they can download everything and perform their own favorite analysis and pull out all the information. So basically, you can download pretty much everything. So you cannot download so far the spaces, because we are still validating some of the results with specifically with the spaces coming from metagenomes. So as I said, so you can download the data. So there are two different ways that you can download the data from. Through the JGI portals. So there is a easy download button at the top. So you can find the URL over here. So you can find this information here, or even at the landing page of the img-vr. So you find these download either the HMMs models that we have for the prediction pipeline, or the complete database with the table with metadata associated for every single contig. So, oops, over here. Yeah. So, how did we obtain the data? So, I mean, where are the data coming from? So, I will summarize the three main points. What is our pipeline for our viral discovery? So how we ended up with a certain manually curated set of viral protein families, and then how we cluster our contigs. Yeah. So basically, our pipeline it's now very, let's say, well defined. In a Nature protocol paper, but as a quick overview, so basically what we did was we used all the isolate viruses at the database in the moment that we performed the study, and we created some models out of them, and we used these models just to fish out of the currently more than over 10,000 different samples, metagenomic samples that we have, and we were able to collect a certain number of metagenomic viral contigs with a certain length across different habitat types. And basically, we use this information to merge with everything from the isolates, just to create a data set of 25,000 viral protein families that we manually curate just to be found mainly or almost exclusively in viruses. And then we use a second round of fishing for which we were able to predict this number of viruses. And then when we have these environmental viruses, so we group with all the isolate viruses as well, and we created these viral OTUs. So currently we have around a little bit more than 100,000 clusters and over 260 singletons. So in our pipeline, so the idea is to for the next steps is to first of all maintain this this pipeline and do this effort periodically over time. The more sequences, samples that we are getting, so the more viruses we are getting with the same sample. But ideally, we'll be combining our pipeline with a VirSorter approach, so we can expand the number of models that VirSorter contain. But this is an automated process, so that will be easier for us as well. Obviously, the database that we have is a DNA viral database. So we want to complement this with RNA viruses coming from metatranscriptomes. This is an ongoing effort that we are doing right now. And then we know that our pipeline is not specifically designed to capture some specific types of viruses, like very tiny single-stranded DNA viruses, some of these viral entities that we'll talk a little bit tomorrow, these virophages, these giant viruses, and so on and so far. And also, ideally, we'll like to add prophage information. So regarding the CRISPR database, so we are using a modified version of the CRISPR recognition tool at the JGI, and basically we are using this CRISPR spacer database for two reasons. The first one is that to predict a host, so we are actually using the information that we get from CRISPRs coming directly from microbial isolates to predict directly a host, and we are also providing information of the spaces coming from all different microbiomes, not for host recognition, that in some cases we are able to predict the host based on these matches when we have a contig that is long enough to have a taxonomic assignment by itself, but also to create these networks with their habitat distribution. Regarding metadata, as I said, so the host prediction is coming exclusively so far from CRISPR spacer isolates and also from the clustering method when we find metagenomic viral contigs clustered with either non-isolates or that they are clustering with metagenomic viral contigs that they have a spacer hit that we can assign as well. So we are expanding the information to all the members of the groups. So ideally, in the next version, we will be adding some other information to predict host. So some of them we already have tested. That is the match of tRNAs that they are present in viral contigs with other tRNAs that they are found in microbial sequences. Also, we are exploring some other approaches like tetranucleotide frequency or the use of some other potential important genes like methylases or lysozymes that they could give us an idea of a prediction of the host. Regarding the type, the habitat type information, so we rely on GOL, the Genome Online Database. So this is so we have a pretty good breakdown of the different levels that you can find. You can, let's say, classify your sample, and also we rely on GOL for the geographic location. So we have the longitude and latitude for the vast majority of all our sample collection. So just to finish, so we started this effort back in August 2016, where we predict a bunch of novel sequences and unique sequences. So that was estimation of 16-fold, whatever was deposited at that time. But then over the last year and a half, so we multiplied these by 6 times. And an important point is that we are not only expanding the numbers, so we are also with these new numbers, so we are also expanding the number of viral sequences that they are predicted to be infecting members of the microbial tree of life. So which every release of our database, so we are predicting for the first time isolates viral contigs assigned to some of these culture or uncultured microbes. So with that, I would like to take any questions, but also I would like to emphasize that this was a major and collective effort from people of this prokaryote super program led by Nikos, Natalia, and Emily, and some new great hires like Simon, Steven, and also many thanks to IMG team, which are the responsible for having the website, maintain the information that we got from the Gold team, and also a bunch of different collaborators, many of them here in the audience that contributed with their samples deposited in IMG for us to mine the viral database. So with that, thank you very much, and I'll take any questions.